Hi, my name is Ashlyn Christiansen, and today I'm going to talk about the anatomy and enzymology of fat absorption. As seen in the right-hand corner, dietary fats come in a variety of different forms. They are found in oils, butter, nuts, avocados, and much more. Most fats are triglycerides, but there are also phospholipids, fat-soluble vitamins, cholesterol, and long fatty acid chains. All of these molecules associate with one another because they are either hydrophobic or amphipathic in the case of phospholipids. This raises an issue for digestion because hydrophobic molecules do not readily associate with water, and the digestive environment is aqueous. This makes the absorption of fat extremely different than the absorption of proteins or carbohydrates. Seen on the right here is the anatomy of the GI tract. As food is ingested, it goes into the oral cavity, makes its way down the esophagus, into the stomach, and then goes through the small intestine and then the large intestine and then out the anus. Food is broken down mechanically and chemically in the mouth, and this is where fat digestion starts. Once it reaches the stomach, little digestion for the fat is done here, but the aqueous chyme solution makes fat form fat droplets. Once in the small intestine, where most of the fat is broken down, bile is excreted from the gallbladder, which you can be seen here, and bile is made in the liver and then transferred to the gallbladder through hepatocytes, and then from the gallbladder to the pancreas through the common bile duct. Bile is a non-enzymatic solution. It contains bile pigment, cholesterol, and most importantly, bile salts. Bile salts emulsify the fat droplets that form in the stomach. Bile salts are reabsorbed in the ileum or changed into bile acids and absorbed in the colon, then brought back to the liver through the hepatic vein. Bile salts cycle through the intestines 2 to 5 times each meal. Fat is then absorbed after it is digested by the enzymes through the intestine's epithelial cells and distributed throughout the body. As can be seen in this diagram, you can see the bile salts surrounding the fat droplets, making them smaller and have an increase in surface area. This is very important for the enzyme action in the small intestine. The enzymes involved in fat digestion include lipase, colipase, and phospholipase. Lipase is found in 2 forms in the body, lingual and pancreatic. Lingual lipase is found in the oral cavity and is the first enzyme to start the breakdown of triglycerides. Pancreatic lipase is made in the pancreas and excreted into the small intestine. Lipase breaks down triglycerides into monoglycerides and 2 fatty acid chains, but it can't do it on its own. Lipase, it cannot break through the bile salt layer to reach the fat droplet, so it needs a cofactor, and this cofactor is colipase. Colipase displaces the bile salts so that lipase can reach the triglycerides. Another important enzyme is phospholipase. Phospholipase breaks down phospholipids in the fat droplet. Colipase and phospholipase are zymogens. This means that they are inactive once they are excreted from the pancreas, and they are activated by trypsin once they are in the small intestine. Lipase, on the other hand, is excreted in its active form, but needs colipase to be fully effective, as seen in this diagram. Colipase has to inject itself into the bile salt layer so that it displaces the bile salts so that lipase can reach the fat droplet and start the breakdown of triglycerides. All of the digested parts and cholesterol that are found in the fat droplets start to coalesce into micelles that make their way down to the bush border of the intestine. A timeline of this digestion is that first fats are ingested, and after mechanical breakdown in the mouth, lingual lipase starts to break down triglycerides. It then goes down to the stomach, where fat droplets form in the aqueous chyme. Little to no fat digestion happens in the stomach. The chyme mixture is then pushed into the small intestine. Bile salts then surround the droplets and emulsify them. This happens because bile salts are amphipathic and allow for interaction between water and phospholipids. They form smaller, more stable fat particles and increase the surface area of fat available to the enzymes, making the digestion more efficient. Then, colipase and lipase are excreted from the pancreas, as well as phospholipase. Colipase, after it is activated, displaces the bile salts, and then lipase can then break down triglycerides, while phospholipases break down the phospholipids. Then the micelles are formed and can make their way to the intestinal wall. The absorption of fat is the next step. In the small intestine, as the micelles reach the bush border, they start to dissolve into their different parts. And the fatty acids and the monoglycerides can move passively through the cell wall, as seen in the bottom here of the diagram. They make their way to the smooth ER to join with and they form back together into triglycerides. Cholesterol, on the other hand, cannot passively diffuse through, and it has to use transport proteins to get through the cell membrane. Once inside, triglycerides, cholesterol, and proteins all form together to make chylomicrons, which can be seen here, which makes its way to the Golgi apparatus. In the Golgi apparatus, the chylomicrons are packaged into vesicles and exported by exocytosis out of the basal lamina. These chylomicrons are very big and cannot fit in through the capillary's basement layer, so they have to go into the lacteals of the lymphatic system, which can be shown here. And they get into the bloodstream to the vena cava right before the blood goes back into the heart. But some shorter fatty acids do not get recruited into chylomicrons and passively diffuse into capillaries and enter the bloodstream that way. This diagram shows a great example of the digestion and absorption of fats in the small intestine. You can see the large fat droplets from the stomach here in the bottom left-hand corner, and then the bile salts come and emulsify them into small droplets, as you can see. Then the micelles are formed after the colipase, lipase, and phospholipase all aid in the digestion of these molecules. They drift down to the bush border, which can be seen by these wavy lines at the apical membrane of the small intestine. And then you can see the two fatty acid chains and the one monoglyceride passively diffusing to the smooth ER, and the cholesterol being transported in. Then you can see the chylomicrons and the Golgi apparatus making these vesicles to exocytose these chylomicrons into the lacteals. The only thing missing would be short fatty acid chains that can passively go through both sides of the cell and then into the capillary. Once in the bloodstream, the fats and cholesterols can be distributed amongst all the body and the cells as needed. And here are my references.